Okay, without further ado, we'll start the afternoon session with Abul, who's going to tell you about T cells.
So yeah, I'm going to make sure you guys don't fall asleep by just warning you up front that if I catch anybody fall asleep, I'm going to shine the laser pointer at them and ask them a question. So you will embarrass yourself in public if you fall asleep. If you think I'm joking, just try me. So yeah, just get rid of that postprandial stupor. One more. I don't know if this is good news or bad news for you, but while I was listening to the morning lectures, I decided I was going to add a few slides which are really basic. And this is because I just want to make sure everybody is on the same page. Really basic slides, and so that's about 10 or 12 that I've added. They're not in your notes. Lori is going to put my slides up on the website so you can access them. The bad news is that because I've added 10 or 12 slides, your break before dinner was an hour long or something. It just got reduced to half an hour. And there's nothing we can do about it. So don't get anxious if my lecture goes beyond 90 minutes. Just relax and sit back, and we'll go on a ride together. And so what I'm going to cover is, and Andy is also going to get antsy because his lecture is the next one. And I always run late, and Andy just starts getting, Andy Lichtman back there, he starts getting antsy because I'm running late. So why don't you all collectively tell him to chill? And so I'm going to start with an introduction to T-cell activation, but I'm also going to revisit the issue of antigen recognition, mostly in vivo, a little bit of basic principles about antigen recognition. Much of the lecture I'm going to spend on co-stimulation. We'll define it. We'll go through the family. A lot of it I will also spend on this third topic, which happens to be one of the most exciting things that's happened in the field in the last 10 years. And then I'm going to show you how you can target both these, the co-stimulators and the inhibitory receptors, for therapy, including some therapeutic strategies that are actually changing the paradigms of immunology. And this is really a cool topic with incredible potential implications. So we're going to constantly bring up sort of therapeutic strategies as we go along. So I want to start with Immunology 101. What do T cells do? And the basic message is very simple. T cells fight cell-associated microbes. They fight microbes that have either been eaten into a phagocyte, and they do it by helping phagocytes to kill better, or they fight microbes that are infecting any old cell. For example, and killer T cells will kill infected cells and eliminate the reservoir of infection. So T cells fight against cell-associated microbes. They also help B cells. I'm not going to spend any time on this population of T cells. This is sort of the T cell, the fashion of the moment, and we are going to come back to the fashion of the moment tomorrow. We're going to talk about I'm losing this. Yeah, I need a... it comes and goes, Mark. Well, I can use this one, but this is a... This is a pretty, I'll use this but you flash it on there and you see, and then it, well.
Is it the batteries?
I don't know. Maybe it's just me. Okay, we'll try. Okay. So we'll talk about regulatory T cells tomorrow. And so the big picture message is that T cells don't see microbes on their own. T cells talk to other cells. And how do you make sure that a T cell will talk to another cell? By enforcing on the T cell receptor one property. It can only see antigens displayed by cell surface molecules. You're with me? So if the antigen receptor can only see antigens displayed by cell surface molecules, it can only function by talking to other cells. A T cell receptor cannot see microbes in the circulation, which antibodies can. What's the cell surface molecule that T cells see antigens displayed on? The major histocompatibility complex, or the HLA. I told you this was Immunology 101, really simple. So that's how you enforce this specificity for other cells, by forcing a T cell to see cell-associated antigen. So what's the life of a T cell? All T cells mature in the thymus. This is the part of the process that we completely ignore in this course. So we're not going to talk about the development of T cells in the thymus. Once they mature in the thymus, they end up in the circulation, and some of them will go into lymph nodes. Jason told you a fair bit about the trafficking of T cells into lymph nodes. I'll come back and revisit this in just one slide in a minute. Here the T cells will see antigens presented by dendritic cells. I'm going to come back and revisit this also in just a minute. These T cells will get activated. Naive T cells are T cells that have not seen antigen, so they are immunologically inexperienced. Just like when you were inexperienced, your parents said you were naive. That's actually how the nomenclature arose. An inexperienced lymphocyte is also called a naive lymphocyte. And so naive lymphocytes will come in here, see antigen, become effector T cells. That term means they're the ones that mediate the effect of the immune response. They do the job. Naive T cells don't do anything. They see antigen, they can't do anything. Effector T cells will do the job of getting rid of the microbe or the infection. So these effector T cells will leave the lymph node, go to the site of infection, and get rid of the microbe. And that, in a nutshell, is the life of a T cell. Now, I'm going to today focus on just this set of events. What happens here? Andy is going to pick up with effector T cells what they are and how they get rid of microbes. So I'm going to ignore this part for now, and we're just going to focus on that. So in order for a T cell, and this is the set of 6 or 8 slides that are not in your notes. And conceptually, it's actually interesting to think it through this way. So in order for T cells to do their job, there's a whole bunch of challenges or problems they have to face. So the first is something that I think Jason alluded to in passing, which is related to the diversity and the specificity of antigen receptors. So what that implies is that there are very few lymphocytes in the body specific for any one antigen. You and I contain about 10 to the 12 lymphocytes, ballpark number. And we estimate that those 10 to the 12 lymphocytes can cover about 1 million or 1 billion specificity, something in that range. And that means that only about 1,000 to lymphocytes can see any one antigen. So the number comes out to about 1 per 100,000 or 1 per million. That's not many. And that small number of lymphocytes has to find the antigen and respond to it. So that's problem number 1. And we're going to go through the problems, and I'll tell you how the immune system has learned how to deal with them. The second is this, that lymphocytes have to be able to locate microbes wherever they enter in your body. Usually they enter through epithelia. So there are 3 ways that you get infections. 1, you touch them. 2, you eat them. And 3, you breathe them. You touch them, eat them, and breathe them. Those are the 3 ways that you get infections. They come in through microbes. It's impossible to think that every T cell of every specificity will constantly be patrolling every epithelium all the time. Can't happen. Not enough T cells in your body. So there has to be a way of bringing the antigen and the T cell together without asking the T cell to be patrolling your gut and your skin and your airways all the time. So how do you bring T cells and antigens together? And we'll see an answer to that. And the third is this problem, that helper T cells help B lymphocytes to make antibodies against circulating microbes or circulating proteins. They help phagocytes to eat stuff in the extracellular environment and destroy it. So helper T cells tend to attack microbes that are floating around and picked up by cells. In contrast, killer T cells or cytotoxic T cells tend to attack microbes that are in the cytoplasm, viruses, or things that have escaped from vesicles into the cytoplasm. So how does the right T cell know what to attack? And so these are the three big challenges that the immune system or the T cells face. And the answer to all three are known. So the way microbes are picked up is illustrated here. This happens to be a skin. This happens to be the example of a skin, but it could be any epithelium. Dendritic cells in the epithelium capture the microbe, and they take it out of the epithelium. Now, when the dendritic cell encounters the microbe triggers what kinds of responses? The early response is the innate immune response. Receptors like TLRs. So the microbe will engage toll-like receptors on this dendritic cell and other receptors, other sensors of innate immunity, and that dendritic cell will get turned on. It'll get activated. One of the consequences of activation of dendritic cells is that it begins to express a chemokine receptor called CCR7. And it's just, Jason mentioned this, but I want to repeat it again because it is truly the most gorgeous story possible. Well, one of the many gorgeous stories. So that dendritic cell begins to express CCR7. And there is only one place where the chemokines that bind to CCR7 are made, and that place is the lymph node in the spleen, the lymphoid organs. It's the only place where the CCR ligands are made. So the ligands are being made here. After seeing a bug, the dendritic cell begins to express the receptor. The dendritic cell gets sucked from the epithelium into the lymph node, carrying its cargo of antigen. So you bring the antigen to the lymph node rather than taking T cells to the site of antigen entry. And through this same region of lymph node are constantly recirculating naive T cells, because in addition to activated dendritic cells, there is only one other population that expresses CCR7. And what's that? What's the population that expresses CCR7? Naive T cells. It has to be that way. And so by forcing receptor expression on the correct cells, you bring them to the correct place. And you bring them to the correct place together. All you're doing is enhancing the chance of a naive T cell finding the antigen. And the process is incredibly efficient because within 12, 18 hours, something like that, if you inject an antigen into a mouse, the mouse will show a detectable T cell response within 12 or 18 hours. So 1,000 T cells in the whole body will find an antigen regardless of where it enters and start making the response. And it's a gorgeously orchestrated process. I just want to emphasize 3 or 4 features of dendritic cells that are worth remembering. They are the most efficient antigen-presenting cells for initiating an immune response, initiating a T cell response. So they are the best stimulators for naive T cells. Why is that? Because they're at the right place. They're located in epithelia. They're also present in all tissues. So if a microbe is present in the tissue, the dendritic cell can pick up the antigen. They have a bunch of receptors for both capturing and reacting to microbes. And I think Ken and others have mentioned some of them, mannose receptors that will capture bugs that have terminal mannose residues on their polysaccharides, toll-like receptors for recognizing, responding to microbes. So dendritic cells have an incredible collection of microbe sensing receptors. They go to the right place. So they're not only present at the right place, they go to the right place, and we just talked about the role of CCR7. And I'm going to tell you in a minute what this means, that they become very good at presenting antigen and stimulating T lymphocytes. And I'll show you in some detail what this means in just a few minutes. So the result of all of this realization that the dendritic cell is really the best cell for presenting antigen, is the tremendous interest in using dendritic cell-based vaccines, so taking a dendritic cell, sticking a tumor antigen in it, and using it to vaccinate against the tumor. To those of you who don't know, this is one of these amazing, amazing stories that's both a happy and a sad story, and it just happened, and it's worth just mentioning it. The story, of course, involves Ralph Steinman, who was a very good friend for many of us and an absolutely wonderful scientist and an even more wonderful human being. Just a remarkable guy. As you know, Ralph got the Nobel Prize for the discovery of dendritic cells. As many of you know, this was also one of those incredibly happy but tragic occasions, because he died 2 days before the prize was announced. He had grown up his own dendritic cells and was using his autologous dendritic cells to vaccinate himself with, because he suffered from pancreatic cancer for about 3 or 4 years. So it's just this amazing story. It's like, you know, it could have been written in a novel or something like that. And it's really I look at it in a very positive way, that it's actually a very happy story, because he got recognized for an extraordinary collection of work. This really is his work. All of this, or much of it, is Ralph's doing. Yeah? They're not at the, so why, the question is, why aren't macrophages as efficient? They're at initiating, remember what I said, at initiating immune responses. They're not at the right place. They don't hang around in epithelia. Macrophages tend not to express as much CCR7, so they don't go, so they don't go to the region where naive T cells are. They don't have as much co-stimulators, and I'm going to show you that. So they're just not that great at turning naive T cells on. Do not go away thinking that macrophages are not APCs. Of course they are. They present antigens to differentiated Th1 cells. That's how you kill a bug. You know, you eat Mycobacterium tuberculosis, a Th1 cell then recognizes the antigen on the macrophage and kills the MTB, right? So so macrophages can be very good antigen-presenting cells, but not for initiating the response, and that's the reason. By the way, just as an aside, so one dendritic cell-based tumor vaccine is now approved. We still aren't totally, the world isn't totally convinced about how good it's going to prove to be. My own bias is that cell-based therapies are very tough to commercialize. It's just too expensive to make them. They're not off-the-shelf things, so we don't know how far Provenge or the currently available prostate cancer dendritic cell-based vaccine, we don't know how far it's going to go, but there is one that is approved based on this. And completing Immunology 101, this is what a T cell is up there. The antigen-presenting cell is down here. This is the MHC molecule. Humans, obviously, it's called HLA. This is what it sees. The MHC molecule's function is to display peptides. The T cell recognizes this complex of MHC and peptide. The most famous crystal structures in immunology are these. This is a class 1, or HLA A, B, and C. This is a class 2 molecule, or HLA DP, DQ, and DR. Class 1 binds CD8 at the tail. Class 2 binds CD4, and the top is where peptides are displayed. And I'm going over this very quickly because all of you should know this, so you really don't need me to spend any time. And just quickly to remind you that cytosolic proteins will go through a pathway that Andrea told you about and be displayed by class 1 molecules. Because CD8 binds class 1, therefore CD8 T cells will see cytosolic antigens, right? Because of this pathway, and CD8 T cells are the ones that functionally become what? CD8 T cells become what? Helpers or killers? cytotoxic. Conversely, CD4 T cells will see vesicular antigens, because the vesicular antigens are displayed by class 2, and CD4 binds class 2. So this is how the third problem that we had raised, that the correct T cell has to recognize the correct type of antigen. That's achieved by segregating the pathways of antigen processing and giving CD4 and CD8 the appropriate binding specificity. There is an interesting question here. And oh, by the way, I show this like this. There are exceptions to the rule, so I'm not going to get into nitpicky exceptions. This is really painting with a very broad brush. There is an interesting question which no immunologist has ever been able to answer. Why does the acquisition of CD4 correlate with helper function, and why does the activation of CD8 correlate with killer function? What's CD4 and CD8 got to do with the machinery of the cell? And we actually don't really know the answer to that question. We sort of know that that's the way it is, but we've never been able to convincingly answer it. Okay. So that was Immunology 101. Now you know how T cells see antigen, where the antigen is picked up, where it's taken, and where you get an immune response going. I'm going to stop at this point and see if anybody has questions. No? Yeah?
When you mentioned the frequency of any T cell for an antigen,
Yes.
were you talking about a particular peptide or
Single peptide. So it's T cell, the number of T cells that recognize any one peptide antigen. It's actually another, since you asked that question, there is another surprise. If you look at least in inbred mice that have only one HLA molecule or one MHC molecule, right? A complex virus might have only 3 peptides that mouse can recognize. So and this is what Andrea was referring to as the concept of immunodominance. There are actually not that many. For any one MHC, even in complex bugs, which has also been a bit of a surprise. But we're not going to go there. Okay, now we're going to start on what I was really supposed to talk about, which is lymphocyte activation. And this is, again, extremely simple notions. So normally the lymphocytes that are floating around in your body that haven't seen antigen, they're in a resting state. They're not doing anything. They rapidly respond to antigen. As I'm going to show you, the response consists of 2 changes. They divide or proliferate, and they differentiate. And I'm going to define differentiation in just a minute. Once they've done that, they migrate to tissues, at least the differentiated ones. Some of them will migrate to tissues where they do their job. And it's become pretty obvious that there are many steps in this process where once you understand the biology, you can target the steps therapeutically. And this has been really one of the most exciting advances in the field. So again, very simple. T cells, naive T cells see antigens displayed by dendritic cells in the lymph node. The dendritic cells are coming from some epithelium. The effector T cells see antigen displayed by any old APC. This could be a macrophage. This could be anything else displaying the antigen, and the effector T cell does its job. Typically, activation of naive T cells has more stringent requirements. It needs more signals. So that's why it relies on dendritic cells, which are the most potent APCs for a number of reasons, which I'm going to explain to you. And it happens in lymphoid organs because that's where the naive T cells go. That's where the dendritic cells bring antigen. So naive T cell activation has a lot more requirements, and the anatomy forces it to be happening in lymph nodes. In contrast, effector T cells have less stringent activation requirements. They can respond to antigen presented by a whole bunch of antigen-presenting cells. And this can happen in any tissue. It is not necessarily constrained to lymphoid organs. So there are real differences between these 2 activation events, although the recognition part, the MHC peptide part, is basically the same. So, but there are real differences in the requirements. And so what are the steps in a T cell response? And we'll just go over the CD4 response. The CD8 response in principle is similar, and Andy will tell you a little bit more about the biology of CD8 cells in the next lecture. So Here's a naive T cell. It sees antigen being displayed by an antigen-presenting cell. One of the first things it does is make the cytokines. One of the first cytokines it makes is interleukin-2. We have always thought of interleukin-2 as T cell growth factor, and tomorrow I'm going to turn all your notions of interleukin-2 on their head and tell you that it doesn't really do what you thought it did, but you're going to just have to wait to hear that until tomorrow. For the moment, interleukin-2 does drive the response. These T cells go into cycle and they proliferate. Immunologists have called that clonal expansion, the idea being that one clone of lymphocytes. In the immune system, we refer to a clone as a cell and its progeny, all of which express the same antigen receptor. So they have the same specificity. So the antigen-specific clone, which may have started with 1,000 cells, becomes a much bigger clone of antigen-specific lymphocytes. And at the end, I'm going to give you an example of the magnitude of that expansion, which is absolutely unbelievable. So that process is called clonal expansion, and some of the progeny of this expanded clone will become effector cells or memory cells, and so that's differentiation. Differentiation, remember, means a change in phenotype. A stem cell becomes a neuron, that's differentiation. A naive cell becomes an effector cell, that's differentiation. So the two big events in lymphocyte activation are proliferation and differentiation. Now, I'm not going to spend much time on differentiation because Andy will go over that for both CD4 and CD8 cells in the next lecture. We're really going to focus on the early events. So what do you need to turn a T cell on? You need three classes of signals. You always need antigen. And I'm going to tell you a little bit about this notion in a minute. You always need antigen. That's why we call antigen signal 1. It's the necessary first signal. And you always need antigen because that's how the response is specific. If you have an influenza virus infection, you want the influenza virus T cells in your body to respond. You don't want hepatitis virus-specific T cells to respond. That happens because you always need antigen recognition to respond. So antigen is the necessary first signal that maintains specificity. Co-stimulators we are going to spend a lot of time on in this lecture, and cytokines I think Andy will cover in much more detail in the next lecture. So, how is antigen recognized? And this is a gorgeous picture of antigen being recognized by a T cell. So here is a piece of the T cell receptor. Again, the T cell would be up through the ceiling. Here's an HLA molecule. The antigen-presenting cell would be down through the floor. And here's a single peptide in yellow being recognized by the T cell receptor. One of the remarkable aspects, one of the remarkable features of this recognition event is that if you look carefully, you'll see that the T cell is actually seeing only 1 or 2 residues of the peptide, which is true. Lots of these crystal structures have been done by now, and the T cell doesn't see much. And so that leads to the quite extraordinary conclusion that you, every day of your life, are making the distinction between influenza virus and herpes virus based on the recognition of 1 or 2 residues, which is sort of amazing and a little bit difficult to understand structurally. There are other constraints that I'm not going into about who's presenting it and so on and so forth. But it's just there's nothing more we can say. It's just what the crystal structure shows you, that you don't recognize a lot to maintain specificity. So T cells are very interesting, regardless of what Jason said when he implied that they just follow B lymphocytes around, they don't do very much. Don't listen to him. They're very interesting because the T cell receptor itself is nowhere near enough to get the response going. You need a bunch of other things. There are teleologic reasons why, and I'm not going to go into it. What are those other things? So first of all, the T cell receptor recognizes MHC peptide. Attached non-covalently to the T cell receptor are several proteins, including proteins of the CD3 complex and the zeta chain, all of which have signaling domains in their cytoplasmic tails. These are the signaling end of the T cell receptor. These three together, T cell receptor, CD3, and zeta chain, together make up the T cell receptor complex. So the TCR recognizes antigen, CD3 and zeta are the business parts. They're the ones that deliver the signals. In addition, CD4 recognizes MHC at the same time as the T cell receptor recognizes peptide MHC. So CD4, and this is a helper T cell. On a killer T cell, it would be CD8. So CD4 and CD8 are called co-receptors. And I really want to emphasize nomenclature because people get very confused about the nomenclature. They're called co-receptors because they recognize a piece of the same thing that the receptor recognizes. With me? And then there are molecules, the prototype of which is CD28, which I want to tell you about, which are called co-stimulators. And that's because they stimulate when they work in conjunction with the antigen receptor. So the antigen receptor stimulates, or the antigen receptor complex, and CD28 co-stimulates. It works together. So co-receptors and co-stimulators are completely different. They both function as signaling molecules. Yup. So it's a very good question. The question is co-stimulation equally important for macrophages and dendritic cells? And the answer is probably not as critical here. Less stringent requirements, various APCs will work, less co-stimulation needed. You know, people ask, is co-stimulation happening or not? I mean, nothing is all or none. There isn't a biological phenomenon where you can say 100% this and 0% this. It's all thresholds. You need a lot more for naive T cells than you need for effector T cells. And that's effector or memory T cells. And that's basically all you can say. So, and I'm going to come back to this in a minute, actually, with a couple more slides. And so, and LFA1 is an integrin. Again, Jason talked about integrins in the context of migration through blood vessels. Integrins also mediate attachments between T cells and APCs. So the integrins are the glues that hold cells together. And so it's again a very pretty system. Some do recognizing, some do signaling, some hold them together. And that's how the system works. All of these have been targeted for therapy. Anti-CD3 is the first monoclonal antibody used in humans. It was used to deplete T cells in patients with acute graft rejection. It was, of course, called OKT3. I think John Maltzman will tell you a little bit more. It's actually surprisingly not used as much now as it was in the past, and there's another version of an anti-T cell antibody that's much more crude than this that seems to be gaining more popularity in the transplant community. But I'm not going to go into that. I think Scott will tell you about targeting integrins for blocking leukocyte infiltration into tissues. Two of the integrins have been targeted. Is the anti-LFA1 now clinically approved, the beta 1? That was the one that was withdrawn. So it was approved and then withdrawn. The anti-VLA4 is Tisabri, and Scott will tell you about it in a minute. And I'm going to tell you much more about co-stimulators. So here's one of the really interesting phenomenon, as you'll gather listening to me, there are lots of really interesting phenomena in immunology. So here's another one that I'll tell you about. So this is what the surface of a T cell looks like, made up of course, and this is what an antigen-presenting cell looks like before the antigen has been recognized. T cell receptors, glues, signaling molecules kind of loosely floating around. Within seconds of antigen recognition, when that happens, these molecules get spectacularly reoriented, such that CD4, not shown here, CD28, the signaling molecules all come to the middle, and the glues go around the periphery. And so if you think about it, what you've just accomplished is create a ring that will hold two cells together, right? Those are the glues, the integrins, and have all the signaling proteins in the middle of the ring, which is where you want them. The central part, this business end, which is doing the signaling, hang on one sec. Let me go through this and I'll come to your question. The central part is called the central supramolecular activation cluster. You don't need to know that. We all call it the CSMAC. And the peripheral integrin-rich ring is the PSMAC, or peripheral supramolecular cluster. And you can see that by fluorescence and so on and so forth. This was all done more than 10 years ago. It's amazing. It's that some signal is telling the right molecule to go to the right place, which is absolutely amazing. There's another even more amazing aspect to this. Somebody is going to tell me what is the most abundant, before you do that. In order to deliver signals to a cell, I'm now being extremely general. What family of enzymes do you tend to turn on in order to deliver activating signals? Kinases. You put phosphate groups on substrates. You tend to turn on kinases. In order to terminate signals, what do you turn on? Phosphatases. What's the most abundant phosphatase in a T cell? Somebody's going to tell me that. So the most abundant phosphatase in a T cell is CD45, once upon a time known as leukocyte common antigen. And so if you want to deliver signals which are going to be dependent on kinases, where do you want the phosphatases? In the middle or outside? In the middle or outside? Outside. So all the CD45 goes over here. It gets excluded from the PSMAC. Integrin, signaling molecules, and the CD45 out. It's amazing. Within a minute, a T cell knows where to put these molecules and stick them in the right place. Borrowing from the neuroscientists, this structure with the space in between is called the immunological synapse. And so you had a question.
Yeah, about the definition of immunology. Is that only TCR recognition of energy or could that be PCR?
So the question is, does the immunological synapse apply only to T cells recognizing peptide MHC? And the answer is probably not. The principles are similar for B cells. We tend to use the term only for T cells. We tend to. But the principles will be similar for B cells also, where the B cell receptor and the signaling molecules will coalesce. Now, before I leave this idea, so this is what we all thought the function of the synapse was. It turns out that another important function is that it is a conduit for the insertion of effector molecules. So for example, just supposing this is a macrophage presenting TB antigen, and the T cell has to put gamma interferon into the macrophage in order to turn the macrophage on, and you're going to hear about this. This is the whole, or the vectorial transport of gamma interferon occurs right through the synapse. That way, the effector molecules get constrained. They act on the right cell. They don't go this way. They go this way as needed. So that's the second function of the immunological synapse. It's now turned out that there are all kinds of inhibitory molecules that also rapidly get recruited to the synapse. So some people are saying it also plays a role in terminating signals. It can start signals, but it can also terminate signals. So it's not really clear how these two are balanced, and I'm not going to get into it anymore. But you guys should understand what the meaning of the term immune synapse is. Yeah.
If some molecules I have seen still called immunological synapse, like if you don't have
That's an interesting question. The question is if CD28, I'm not even showing that here, but if CD28 is not present, should you still call it an immunological synapse? Probably yes, but I'm not going to go. That's really getting into a subtlety. So what are the minimal requirements for calling something? In my mind, it's the T cell receptor and the co-receptor with the integrins on the outside. That's why that's all we show in the book. We don't even show the co-receptor. But that's just me. I mean, the aficionados in the field may have much more strict definitions. Yeah? Yeah. No, so cytokine. So the question is, are the effector molecules that are delivered to the APC all preformed? And cytokines aren't preformed. They're rapidly synthesized upon activation. So many of them will be rapidly synthesized. Others might be in perforin. For a killer T cell, if this was a CTL, perforin and granzymes are preformed and sitting in the granules, and this is the way the granules go. So it depends. Let's move on. Now, Art Weiss, who's my close friend at UCSF and one of the world's experts on T-cell signaling, gets totally horrified when I give a lecture on T-cells, because this is the only slide on signaling you're ever going to see from me. This is an enormous field. Thousands of people have worked on lymphocyte signaling, and I'm embarrassed to admit that we're basically going to blow it off. And so just don't email my buddy Art Weiss and tell him what I just did. But I just want to emphasize general principles. The principles of signaling are that there's some signal from the outside that leads to the production of biochemical intermediates, enzymes, kinases, what have you, that ultimately lead to the activation of transcription factors. And that's kind of the punchline. And I'm listing 4 major pathways which are true, but there is one thing really wrong about this picture, about this list. I'm listing these pretending that they are linear pathways, and they're really not. If you listen to the signaling mavens, they'll talk to you about networks and hubs and stuff like that. So that's when I say, time to go to the next slide. No, I'm just kidding. You should have a much more sophisticated understanding of lymphocyte signaling than what I'm telling you. That's all I'm going to say, and I'm going to leave it at that. And that's a cytokine signaling pathway that I think Andy is going to cover later on. Okay, now we get to what I'm really interested in. God, it took only 45 minutes, Andy. And now we're getting to the business end of our lecture. But anyway, so this is a really interesting story. So this story, in a most big picture way, is this, which is the idea. Remember, the immune system does only one useful thing. What does the immune system do? It does only one useful thing. What does it do? It does what? It fights infections. That's the only real useful thing that the immune system does. You don't have an immune system, you drop dead of infections sooner or later. And so that's the one useful thing it does. And so the system needs a way of saying, I need to respond to an infection, and I do not need to respond to all the harmless junk that I encounter every day of my life. And so that leads to the concept that in order to turn on a lymphocyte, you need 2 signals. Signal 1 is always antigen, and signal 2 is something that is induced by the innate immune response to microbes. innate immunity occurs. I'm going to say it, and then I'm going to qualify that statement. Innate immunity is designed to occur against microbes. Therefore, because that's a requirement, you respond preferentially to microbes. Now, do you respond to transplants? Yes or no? Where's the microbe? Do you respond to tumors? Yes or no? Where's the second signal? So it turns out, and I missed part of Ken's lecture, but this is his favorite topic, so I'm sure he covered it, that in addition to microbes, the innate immune system responds to the products of dead and damaged cells. This is how you clear junk. And it is postulated that transplants in microbes have enough dead cells that they elicit the same reaction. It's the dead cells that are providing signal 2 rather than microbes. So those are the, quote, exceptions to the rule. They're really not exceptions. It's just the other stimuli that give you innate immunity. So signal 1 plus signal 2, and now the cell takes off. If the cell gets only signal 2, it cannot respond. That's the principle. So if you look in a little more detail at a T cell, here's a T cell that has not seen a bug. It might be presenting an antigen, and we are faking this, of course. What would the antigen be? Anything. Here's a naive T cell. The naive T cell has the T cell receptor in the business end. It also has this molecule called CD28. There is nothing to engage CD28. So this T cell gets signal 1. Does it get signal 2? Yes or no? No. It cannot respond. And don't worry about this energy. We're going to go over that tomorrow. It cannot respond because it did not get all the necessary signals. Now that antigen-presenting cell or the dendritic cell sees a microbe, and one of the things that happens during the innate immune response is in response to the usual sensors, TLRs, etc., the dendritic cell puts on its surface a whole bunch of membrane proteins, the prototype of which is a protein called B7. And it's called B7 because during a search for molecules expressed on B cells, this was the 7th one found. It ain't got a whole lot to do with B cells. You know, immunologists use idiotic nomenclature. So I'll tell you another example of idiotic nomenclature in a minute. So this was called B7. As you know, there are 2 B7s, and the formal name is CD80 and CD86, and I'm going to come to that in a minute. Turns out that CD28 is a receptor for B7. So now this dendritic cell saw a microbe, made an innate immune response, put B7 on its surface. Now you engage CD28, signal 1, signal 2, the T cell takes off. That's the 2-signal concept, that you need second signals that are provided mostly by microbes, possibly by dead cells, dead and damaged cells. With me so far? So life was very simple at this time. Until, so this is just in words, and I'm not going to go, or I've already told you a little bit about this. I'm not going to go over what co-stimulation is. Life was very simple until the molecular biologists started asking, well, how many homologs of B7 are there in the genome, and how many homologs of CD28? And you know, you let the molecular biologists loose, and the first thing they do is create confusion. So they big time created confusion. And I joke with my molecular biology friends that they created the confusion, the rest of us had to go back and figure it all out. There is some truth to this, but I'm just joking, obviously. So it turns out that there are 6 or 7 B7 homologs, and there are 4 CD28 homologs. And these are the names, and this is in your notes. I'm going to pick just a few examples of these in a minute. So that's story number 1, that it's not just B7 and CD28, that there's a family of B7s and a family of CD28 or its cousins. But the even more interesting story is that they don't all do the same thing. B7 CD28 interactions are particularly important for getting immune responses going. So this is the original definition of co-stimulation. It's the signals you need to activate a naive T cell in addition to antigen. They are the prototype. This is a pair. ICOS stands for inducible co-stimulator, and Shiv will tell you there are lots of functions that have been described. Its most important function seems to be in germinal center B cell reactions, and Shiv will tell you more about that. But what's even more interesting than the existence of multiple family members is that at least two of them don't turn cells on, they turn cells off. So one is CTLA4, so-called because in a search for antigens on cytotoxic T lymphocytes, this was the fourth one discovered, CTL antigen number 4, it ain't got nothing to do with cytotoxic T lymphocytes. It's an inhibitory receptor and a member of the of the CD28 family. And a second one called PD1, and I'm going to tell you all of this, more about these in just a few minutes. So there is an obvious problem here, nomenclature problem. Is CTLA4 a co-stimulator? Yes or no? Yes or no? Yes. It inhibits. So what are you going to call it? And you got a problem. And so it turns out, just think about it. CTLA4 and PD1 are members of the family, same family, but they inhibit, they don't activate, they don't stimulate. So what are you going to call them? So people have gotten into all kinds of conniptions over this. I mean, some call them co-inhibitors, and some call them this and that. And I'm not going to take a stand on this issue. I just want you to understand that you can't call them co-stimulators, because they don't co-stimulate. And so my preference is to just call them CTLA4 and PD1 and just not get into this nomenclature mess. Just call it CD28 and ICOS, and don't try to lump things together into families when the lumping doesn't work anymore. But all of us immunologists have a great fondness for lumping, and this is one situation where you'll get into trouble if you try. So just be aware of the problem. You will also see in the literature the term co-inhibitors. And this is what they're referring to. They're referring really to CTLA4, PD1, and a few others that I'm going to just mention in passing later on. And so how do we know co-stimulators are important? This just happens to be a 15-year-old experiment from our lab. All of you know about CFSE dye dilution analyses? Yes or no? So in the old days, we used to do, if you wanted to measure proliferation, what was the assay you would do? Thymidine incorporation, right? You'd take a culture, you'd pulse it with tritiated thymidine, and you'd measure how much thymidine got incorporated into nuclear DNA. Well, first of all, that's an assay not just for proliferation, but also for survival. The more the surviving cells, the more thymidine. Second, you can't do it in vivo. There's no way of following thymidine incorporation in cells in vivo. So the assay that has been developed, and all of you should know this, is an assay that uses a fluorescent dye called CFSE. In case you wondered, that stands for carboxyfluorescein succinimide ester. It esterifies proteins in the cytoplasm of cells, so the whole cell begins to glow green. And a whole bunch of cytoplasmic proteins are esterified. Every time a population of cells divides, the amount of fluorescent dye is reduced by half. So if you take wild-type T cells and expose them to antigen, the cells will go through multiple divisions. If you take T cells that are exposed to antigen in the absence of B7, they won't divide. The majority of them will stay undivided. This is the kind of experiment that says that you need co-stimulation for T cells to take off. Again, the details don't matter. This is now the preferred method for looking at cycling of cells in vitro. You can also put these cells in vivo and follow them and so on and so forth. So all of you should understand, one of the asides of this course, and I didn't make this point earlier, is one of the things we try to do is to make you more comfortable reading the immunology literature. And that's why I tend to emphasize some of these points. So you should all understand what CFSE dye dilution means. And so this is a simple experiment that just says cells need B7 to divide. But this problem too has gotten complicated. After I went through all that song and dance about CFSE and its value, the next slide I'm going to show you uses thymidine incorporation. So it's actually a very old experiment. It's actually done in the days when Ken Rock and I used to collaborate on experiments. And so this says that naive T cells will divide when the antigen-presenting cells are wild type, but if the antigen-presenting cells lack B7, they won't divide. But if you take memory cells, they'll divide a lot and very fast with normal APCs, antigen-presenting cells, but they'll even divide pretty well with B7 deficient APCs. So this kind of experiment, lots of people have done these, say that memory cells are less co-stimulation dependent than naive cells, right? So co-stimulation has a very big impact on naive cells, less of an impact on memory cells. And I make this point because if you guys go back and do experiments and you block co-stimulators and you don't see an effect, that may be because you're looking at all memory cells. So you have to be careful what your starting population is if you're going to interpret these experiments. And then my friend Jeff Bluestone, his lab, when Jeff was still in Chicago, did an experiment that came as an even bigger surprise. So what Jeff was interested in was, well, it was a simple hypothesis. There's a model of type 1 diabetes called the non-obese diabetic mouse, or the NOD mouse. And this mouse makes a T-cell attack against islets, destroys its islets, and it's all spontaneous. So Jeff said if you get rid of co-stimulation, the T cells won't respond, correct? Yes? So will you get reduced diabetes? Yes? Yes. If you get rid of co-stimulation, you won't get diabetes, correct? With me? So there's a lesson you're going to learn from me today and tomorrow. If all our predictions were right, immunology would be incredibly dull. And the one thing it is not is incredibly dull. In fact, it's incredibly cool because Jeff's lab made the prediction. This is a wild-type NOD mouse in dashed lines. This is the incidence of diabetes. You take away co-stimulation and you get much worse disease. Right? Right? That's the data. And so why do you get much worse disease? This was a huge puzzle when the result came out. And the answer is that regulatory T cells, which are present in a normal mouse, practically disappear in a B7 knockout. So regulatory T cells are also B7 co-stimulation dependent, just the maintenance. And what happened in that experiment, in the Bluestone Labs experiment, is that the Tregs, the regulatory T cells, disappeared, but some T cell population that didn't need a lot of co-stimulation attacked the islets. We still don't know what that population was, and that's a 15-year-old experiment. So, but the regulatory cells. So now you come to the conclusion that you absolutely need co-stimulation to initiate responses, and that's B7 CD28, but different populations vary in their dependence on the B7 CD28 pathway. Typically, naive more than activated more than memory, dependence. CD4s more than CD8s. In fact, in those NOD experiments, the thinking is that islets are being destroyed by CD8s, which are not so B7 dependent. And regulatory T cells are B7. So it's not a simple answer that you need B7 to turn on T cells. Yes, you do, but depending on which T cells you're looking at, which populations, at what time. And it's just important to remember this complexity to interpret your own experiments. That's really the message I want to get across. Oh, this is so one of the problems we don't know the answer to, does CD28 signal by itself and work in conjunction with T cell receptor signals? or does CD28 make the T cell receptor signal better? So are there really two pathways, or is CD28 really amplifying the signaling pathway from the TCR complex? And amazingly, if you look at the literature, you'll get both answers. And I'm not going to go to the consequences of CD28 signaling. This is another area that's been a huge black box. It's been very tough to nail down. This is why there are so far no small molecule inhibitors of the CD28 pathway. Zero. None. We can't figure it out. And so this remains a big hole in the field. There are other unresolved questions. So we know a lot about the B7 CD28 pathway, and I've showed you some of that. But there are many other unresolved questions. So are B7-1 and B7-2 the same? My own bias is yes, because working with Arlene Sharp, who knocked them out, if you look at the B7-1 or the B7-2 knockout mouse, the individual knockouts, they make pretty good responses. If you knock both out, the response goes away completely. So that implies that they are functionally redundant. But you will read the literature, you will find papers saying they're not quite the same. They have subtle differences. So I'm just leaving you with unanswered questions. My bias is that they're functionally redundant, but not everybody will agree with me. Do B7 molecules signal backwards? So far I've talked, if you just think about it, here's the APC, the dendritic cell, that expresses B7. Here's the T cell that expresses what receptor? CD28, and the signal goes like that. APC to T cell, correct? So does the signal go backwards? And there are published papers that they say that the signal goes backwards. There has never been any biochemistry to verify this. So I'm just acknowledging biases. I tend to view the world a little more black and white, so I say no. But just go away with the statement that there are published papers out there that B7 can signal backwards into the APCs. I don't think it's convincing data, but it's still out there. So these are these unresolved questions that I'm telling you about that are still out there in the field. Yeah? So it's a good question she asked. She said, signaling backwards doing what? And I really didn't want to get into this because it's signaling backwards and making the APC an inhibitory APC and ta-da. You know, it's complicated and very little biochemistry, complex biology, most of it done in vitro. Take it for what it is. You shouldn't dismiss anybody's papers. You know, you should pay attention to anything that's published. Take it for what it is. And so that's where the field is at the moment. Yeah? Is CD28 exclusively expressed by T cells? As I said a minute ago, nothing in biology is 100%. So plasma cells express low levels of CD28. What on earth is it doing on plasma cells? I have no clue. So it's out there. There may be other populations. For our purpose, it's expressed only on T cells. There's another interesting story. In humans, but not in mice, especially as humans age, about half the CD8 T cells tend to lose CD28. We don't know why. We don't know if that has a functional consequence. This is all stuff that's out there.
Is there any other redundancy between the CD7 family or the CD28 family?
Is there any other redundancy? Let's come back to that question when I tell you about inhibitory receptors. We'll come back to that. Remember to ask me. So I just want to be clear. I think this is redundant, but don't go away thinking that's gospel. There are people out there who disagree with me. I want to just quickly touch upon this because some of you, especially in the companies, are aware of a rather unfortunate incident that happened about 5 years ago. And there's a group in Germany and then a company that was established in Germany that made an anti-CD28 antibody that they called a superagonist, who was a very strong activator. In fact, it was so strong that it worked even without signal 1. So it violated the rules of immunology. Signal 2 without signal 1 should never work. This one did. And there was some mouse data that said, remember I said B7 is required for regulatory T cells, because regulatory T cells have CD28. There was some mouse data that this antibody was very good at activating regulatory T cells. And so the company put it into a clinical trial. The hope was to use it for patients with rheumatoid arthritis. The clinical trial was a safety trial. It was an extremely poorly planned trial. I'm not going to beat up on the company, especially because it doesn't exist anymore. Yeah, I mean, it's a moot point, right? But what happened was, instead of preferentially activating regulatory T cells, all the T cells in those patients got activated. They got a vicious cytokine storm. One of them almost died. 2 of them have permanent, they basically got a septic shock because they were flooded with cytokines. And the clinicians in the audience will realize they ended up with what's called disseminated intravascular coagulation. Some of them lost fingers and toes. Some of them have permanent CNS deficits because they probably got mini strokes because they clotted off their blood vessels. So it was an unmitigated disaster. Yeah. It was, the trial was for the Brits in the audience. The trial was actually done on 6 healthy 20-some-year-old men at Northwick Park Hospital. It was a bad deal for everyone involved. The interesting and troublesome question that all of you have to ask is this: why were we unable to predict this from the preclinical studies? That's, to me, the most important question. And that's not good enough. Then if you buy that, if you say that that's the answer, then you should never do any preclinical study, ever. But mice aren't people. The important question is you've got to be able to predict what the differences are that account for these sorts of results. And we unfortunately still don't know the answer. Because the company went belly up, not many people are interested in following this up. It's actually a very important problem. All of you in this room are interested in human disease. All of you are interested in human therapeutics. All of you are going to have to use preclinical models to be predictive, and you have to be, don't just say it can't be done, figure out what the problems are and how we make preclinical models as close as possible. So there are all kinds of explanations, but just remember that this is one where our predictive value completely failed. The second problem is this. This is an agonist. It's an activator. And it's dangerous business putting agonists into people. And this was one of the lessons that came out, that you have to be really, really careful. It's much easier to put blockers into people than to put stimulators into people. So you have to be really careful using agonist reagents. No more about this unfortunate clinical trial. You guys can read about it yourselves. Yeah.
Why would they even predict that a stimulator would have been effective in a disease in which the immune system is overactive?
Because why would they have predicted that it would be effective in an autoimmune disease? Because the mouse data said it preferentially activated regulatory T cells. It was not stupid based on the available information. It really wasn't. The people who were involved in the company are not fools. They knew what they were doing. It was just that our predictive capacity failed. So that's the challenging problem. Okay, yeah.
Was that reagent tested on primates?
Yes, it was. But the question is, was it tested on primates? There are a lot of tragedies with this story. One of the tragedies is that the primate data have never been made public. And since the company went belly up, nobody is ever going to make them public. So none of us has seen it. The website of the company said that there were no toxic effects in primates. But you haven't seen the data. Take it for what it is. You either accept the website or you say, I need to see the data before I accept the website. I don't have an answer. I'm not being skeptical. I'm just saying that's exactly what the fact is. That somebody saw the data and accepted it. How much of it? Well, none of us is privy to that. So that's where it stands. That's the whole story, really. That's what is known. And there are some troubling lessons, but it's not worth going into them anymore. Yeah?
Is the
Super agonist.
yes, a super agonist that occurs with infection like
Oh, no, you're talking about super antigens.
is that anything like
No. Super antigens are called that because they bind to the T cell receptor of multiple specificities. So they function like antigens, but they function like antigens on multiple specificities. So it's completely different. Yeah?
So in this case, all the T cells were stimulated. That's what happened usually. That's why.
There was no what?
There was no preferential.
In this case, a lot of the T cells were stimulated. There was not preferential activation of regulatory T cells. That's correct. Okay. Now, so far it's really just been warming up because what my lab really works on is this. So, now we get to the really fun stuff. So this is one of the most interesting stories that has developed. And I want to introduce it with three sort of big picture statements, and then I'm going to burrow down a little more deeply with a couple of examples. So using one way of, and this is something I'm going to emphasize tomorrow. You and I, our immune systems live in a constant state of balance. You have to activate, otherwise you're dead. But you also have to control, otherwise you're dead. You know, in one case you're dead because of infection, the other case you're dead because of autoimmune disease. So you have to live in this balance. You can't let anything go too far. One way, and there are multiple ways you do this, one way you live in this balance is by using different receptors, some to activate and some to inhibit. And I'll show you a gorgeous example of this, really gorgeous example. So these inhibitory receptors have now been described on many of the cells of the immune system. Unfortunately, we don't have an NK cell lecture in this, Scott, correct? There isn't a talk on NK. Oh, Louis is coming tomorrow? Oh, perfect. So Louis Lanier will come tomorrow and he'll tell you about NK cells. And this is one of the prototypes of inhibitory receptors. I'll give you some examples on T cells. You can tell Louis that I thought that T cells are far more interesting, by the way. He's also at UCSF and a very good friend. And this is, again, a very general, big picture concept that many of the activators work by recruiting kinases, and they use signaling domains called ITAMs for immunoreceptor tyrosine-based activation motifs, whereas the inhibitory receptors recruit phosphatases via immunotyrosine-based inhibitory motifs. So ITAMs and ITIMs. These are all big picture statements. So the prototype of the inhibitory receptor in the world of T cells is this, which is that, and there are many, many messages to come through from here. Remember the same old, T cell receptor sees antigen, signal 1, CD28 sees B7, signal 2, the two work together, you get a good response. Yes? So far, so good? Turns out that there's a second receptor for B7, and that's CTLA4. And when it sees the same B7, it shuts the response down. And the importance of this pathway was illustrated by two labs knocking out CTLA4 in mice. And when they knocked out CTLA4, all hell broke loose. 100% of the mice dropped dead of systemic inflammatory disease within 3 or 4 weeks. Now, this is a very informative experiment. It says you don't have to vaccinate. You don't have to change TLR signals. You don't have to do anything to the mouse. All you do is remove one inhibitory receptor and all hell breaks loose. That says that to the extent you are you and I are like mice. We are constantly trying to see self-antigens, and we are constantly controlling the response, right? So self-recognition is happening all the time. It has to be actively controlled. And that alone is an extremely valuable message to get across. It came across the most loud and clear with CTLA-4, subsequently many other examples of this. And Jim Allison's lab 15 years ago, almost 15 years ago now, said if that happens with self-antigens, it controls responses to self, then maybe it controls responses to tumors also. And so what Jim's lab did is this experiment. This is a mouse melanoma transplantation experiment. So if you transplant the melanoma and watch it grow, it'll grow in the mouse till you have to sacrifice the mouse. If you treat the mouse with a course of antibodies that block CTLA-4, if you do it early in the course or even after the tumor has been established, you blow the tumor away. So the tumor is somehow using the CTLA-4 pathway to inhibit immune responses against itself. You have not vaccinated. You've done nothing to the mouse. You just blocked CTLA-4. With me so far? So inhibitory receptors are incredibly powerful. So this is just repeating what I just told you, and I'm not going to go over it in detail. So how does CTLA-4 work? And we all thought, that's why I'm calling it the original hypothesis, because I'm also going to suggest at least partly turning it on its head. We all thought that B7 and CD28 gave positive signals, the usual kinases, and that gave a response. But when CTLA-4 was engaged, it gave some sort of a negative signal, perhaps a phosphatase, and it shut off the response. So this is cell intrinsic. With me? The T cell that sees antigen is also the T cell that's getting shut off. The responding T cell is getting shut off. With me? Cell intrinsic signaling block. So far, so good. Then people start doing experiments and make a mess of the field. And the first experiment, which was really cool, was this. If you take some CTLA-4 deficient T cells and some normal T cells and mix them together and put them into take bone marrow precursors from wild type and CTLA-4 knockout, mix them and put them together in the mouse, right? If the defect is cell intrinsic, then those CTLA-4 knockouts should take off. Yes? There's a cell intrinsic defect. In fact, that mixture behaves like it's normal. Nothing happens. Mouse is completely normal. That alone says it can't be a cell intrinsic defect because it can be controlled by normal T cells. You got to follow the reasoning with me. Yes? Normal T cells can control a CTLA-4 deficient T cell. Yes? So the question is, in this bone marrow chimera, did the CTLA-4 deficient T cells even proliferate in vivo? Nothing. Just like normal. Nothing. I'm going to show you the second half. Then I'm going to take you through a really interesting conundrum. Yes?
Following the second signal, what happened to the expression of the CTLA?
Hold on. Hold on to that question. I'm going to come back to that. And then there was a second series of experiments that said, not only is CTLA-4 expressed on Tregs, on regulatory T cells, which I'm going to tell you about tomorrow, but if you knock it out on regulatory T cells, all hell breaks loose. Selective depletion of CTLA-4. So that led to the conclusion that really what CTLA-4 does is it mediates the suppressive function of regulatory T cells. Because if you take it away, that's the only molecule you've taken away from regulatory T cells, You completely lose control. With me? And that, of course, will explain what happened in the chimera. The regulatory T cells in the knockout mouse were not working, but there were plenty of good regulatory T cells in the wild-type mouse that provided the control. So that experiment says that the real function of CTLA-4 is cell extrinsic. It's coming from the regulatory T cells. Now you got another problem. If it's an inhibitory receptor that transduces inhibitory signals, if you inhibit a regulatory T cell, it should become a lousy regulatory T cell. In fact, it's required for the function of Tregs. Doesn't make sense. Are you following with me? This is a fairly complex argument I'm going through. It's required for the function of regulatory T cells. It can't just be an inhibitory receptor. So my good friend David Sansom, who is at the University of Birmingham, published a paper last year that is one of the most interesting papers I've seen. It's a paper in Science, and I can tell you about it. And I won't go through all the data. What David suggests is that really what CTLA-4 does, it binds to B7 on APCs, and it eats the B7. It strips the B7 off of the antigen-presenting cells. And so a regulatory T cell expresses CTLA-4. It binds to B7 on the dendritic cells. It strips off the B7. And now the dendritic cell can or cannot provide signal 2. It cannot provide signal 2 anymore because it ain't got any B7, and the whole response shuts down. You take away CTLA-4, the B7 is constantly expressed, you constantly provide signal 2, the response is out of control. So CTLA-4 titrates the amount of available B7. This phenomenon, hang on one sec, this phenomenon David called transendocytosis. It's a very sensible explanation for the data that are available. It is a completely unique mechanism for a receptor to function by removing a ligand. It's very interesting. Don't take it as gospel, but just think it through yourself. I think this is the only way that CTLA-4 on the regulatory T cells could possibly work. But that's just my bias. He's a very good friend of mine, so. Yes? It could be a case of, absolutely, it could be that CTLA-4 competes for B7, and I'm going to come to that, except that the data using very pretty confocal microscopy is that you can find complexes of B7 and CTLA-4 in vesicles in the T cell. That's what's so cool. And if the T cell is from a CTLA-4 knockout mouse, you do not find those vesicles. That's what's in the paper. Now you're pushing the extra piece of data. It could be competition, but what he suggested is that it's actually transendocytosis. Yeah?
Going back, is this exactly the same as
Well, is the function of CTLA-4 the same in humans as in mice? And I'm going to show you the reason why we think the answer is yes. Is the mechanism of action the same? How are you going to do this? I mean, this isn't easy to do. So I think people are trying and we don't know. The function of CTLA-4 is probably exactly the same in humans as in mice. I'm going to show you the data for that in a minute. Somebody else had a question. Yeah.
So after activation made naive T cells,
Yes.
it's proliferated and differentiated. Through that process, one of the clones gained the CTLA.
No, so it's not just one clone. I'm going to go into regulatory T cells tomorrow. So we'll come back to that. But actually, many responding T cells can also express CTLA-4. And they might work by exactly the same mechanism. They just strip away the B7 and shut the response down. It's exactly the same. It doesn't matter which cell expresses CTLA-4. It can have exactly the same function. Exactly the same mechanism of action. Yeah?
Is there an error on the first drawing there instead of the CD creation be on the T cell, not on the...
Oh, yeah, those are flipped. Yeah, my fault. Thank you. Yeah.
I was just going to ask, with it looks like you're saying you don't need the signal 1 for the CTLA4 to remove.
You're getting much more complicated now. The question is, do you need signal 1 for the CTLA4 to remove B7? That's getting much more complicated. That's pushing it beyond where it's at. So I just want to, so this is what we think. CTLA4 limits the activation of responding T cells. It mediates the suppressive function of regulatory T cells. It still leaves this conundrum. How does a T cell decide I'm going to use CD28 to recognize B7 and get turned on, or I'm going to use CTLA4 to recognize the same B7 and get turned off? How do you decide? Because that's going to determine the outcome of the response. Correct? So you guys are going to, I'm going to take you through and you're going to answer the question. CTLA4 is most important for preventing responses to self-antigens. Yes? You buy that? And for a minute, let's not worry about which cell is expressing it. Just buy that. When you're are you presenting self-antigens now? Yes or no? Yes. You're not nasty infected. Well, I hope you're not. So you're not nasty infected, so the level of B7 on your dendritic cells is high or low? Low. Big picture statements? Low. You're presenting self-antigen, B7 is low. It turns out that CTLA4 has a 50-fold higher affinity for B7 than does CD28. So which receptor will you preferentially engage? CTLA4. Now you have a bug. The levels of B7 go up, and now you start engaging the low affinity receptor. All it is a balance. When the levels of B7 are low, you preferentially engage the high affinity CTLA4. When B7 levels go up, you preferentially engage the lower affinity, or you can start engaging the lower affinity CD28, and you change the balance. And you guys have to admit that this is one of the most gorgeous control pathways. You fix the affinity of the receptor, so the activating receptor is a low affinity, the inhibitory receptor is high, and you regulate the level of ligand so that low level is with self and high level is with microbe. Can't be any more beautiful than that, yes? Please say yes. Thank you. So it is a really lovely story. Now, oh, don't push it. I mean, you know, yes. This is, I'm actually, now having said all of that, I should, in the spirit of complete truthfulness, I should say that I'm actually pushing the theory a little bit beyond where the data are. But this is really how people are interpreting the results. Do we really know? We do know CTLA4 is a high affinity receptor. I mean, you know, a lot of this is a very interesting idea. But I thought you guys should know a very interesting idea that's emerging. Yes?
But then what happens with the T cells losing CD28?
Yeah, so this is the comment I made. What about T cells losing CD28? I have no idea. They probably won't work at all. So if you don't have CD28, you can't get activated. So there's really nothing to stop.
But will they have the same amount of...
I have no idea. I really have no idea. Those are not so easy to do. Yeah. It's a very good question. So CTLA4, isn't CD28 constitutively expressed? Yes. Isn't CTLA4 induced? Yes, on responding T cells. But there's one cell population where it's constitutively expressed, and that is the regulatory T cells. All regular FOXP3 positive regulatory T cells are CTLA4 high. Okay. So the second inhibitory pathway, and then, Andy, I won't be that late. I was just another 5-10 minutes. What? Shiv. So this is the second inhibitory pathway. It's called PD-1. PD-1 stands for programmed death, because when this receptor was cloned, people thought it was involved in programmed cell death or apoptosis. You had a question. Oh, yeah, you had a human CTLA-4 question. I'm going to come back to that. Programmed death, it turns out it's got very little to do with apoptosis. It's really a homologue of the CD28 family, and it too is an inhibitory receptor. And the same sort of data. If you knock it out, you get autoimmune disease. Just as an aside, the autoimmune disease is much less severe than in the CTLA-4 knockout. The CTLA-4 knockout drops dead, regardless of the strain of mouse. The PD-1 knockout gets much milder autoimmune disease. And I'm going to tell you why that's not a trivial statement in a few minutes. And so Rafi Ahmed's lab did an experiment about 5 years ago, which is another really lovely experiment. They infected a mouse with a virus. It's a mouse virus. It's called lymphocytic choriomeningitis virus. Everybody who's at Emory knows everything there is to know about this virus, because it's popularly used in the Emory Vaccine Center. And so they infected the mouse with virus. UNTX means untreated. If you look at the untreated mouse, it has not many gamma interferon producing cells, pretty lousy CTL activity. That's what this shows, this bar shows, and lots of virus. So virus has not been cleared from the spleen. If you treat this mouse with a single dose of an antibody that blocks the PD-1 pathway, it really was one dose, that mouse gets a big increase in the T-cell response, both cytokine production and CTL activity, and it clears the virus. That experiment says that the chronic virus is somehow using the PD-1 pathway to limit responses against itself, against the virus. Now, we know much less about PD-1 signaling, what it does and so on, much less than we do about CTLA-4. So that's really, I'm not going to get into any of the details I got into with CTLA-4. I just want to summarize. This is what we think these inhibitory receptors do. CTLA-4 and PD-1 are the prototypes. They maintain tolerance to self, and they're involved in immune evasion in tumors and chronic infections and so on. Why so many inhibitory receptors? Just why the redundancy? And somebody had asked me the question. The current concept, evolving concept, is that they do different things. That CTLA-4 is really important in controlling the initial activation in the lymph node, which sort of makes sense, right? If it's working by down-regulating B7, that's where you need the B7. It's the initial activation in the lymph node. Whereas PD-1 may have a more important role in the effector response, especially in CD8 cells. Nothing is all or none. So it's not 100% this and 100% that. It's preferentially. CTLA-4 acts early to control responses, early during lymphocyte activation. PD-1 acts late. So can you exploit these pathways for therapy? And that's where I'm going to end up. Some of it is now pretty well established. So this is the first idea. This is the idea of blocking co-stimulation. Oh, it's actually an interesting story just to walk you through it. So people once said if you block B7, you'll shut the response down, correct? So you make an antibody against B7-1, and you treat someone with that antibody. Will it work, yes or no? No. Why not? Because you got the second family member. So you say, I got B7 too, right? So you say, let's make 2 antibodies. Will the FDA allow you to treat someone with 2 unknown antibodies for the first trial? No. You can't combine them. So you use the TNF receptor approach. You take a receptor protein. Now, the first receptor protein that was tried was CD28. Does it work? Does it work? No, because I just told you, because it is a low affinity receptor. So you take the high affinity receptor, which is CTLA4. I'm just going through the reasoning that people follow. So you take the high affinity receptor. If I inject the extracellular domain of the receptor into you, what's the half-life? Minutes, days, weeks? Minutes. So if you want to increase the half-life, you attach an immunoglobulin tail to it. And Shiv will tell you tomorrow why IgG has a longer half-life than any other circulating protein. And so this is the rationale for making fusion proteins, that you attach an immunoglobulin tail, and now the CTLA4 has the half-life of IgG. With me? And so that's the extracellular domain of CTLA4, not a signaling molecule, with the tail of IgG. That's CTLA4-Ig. I just want everybody to understand, CTLA4-Ig is not anti-CTLA4 antibody. With me? Do not ever make that mistake. CTLA4-Ig is a fusion protein, the extracellular domain of the high affinity receptor for B7. It is functionally the same as anti-B7. Yes? Except it'll bind to both B7s. So it binds to both B7s. It shuts off signaling via CD28. No signal 2, therefore the whole response goes away. That's now approved for the treatment of several inflammatory diseases and for graft rejection. The one that's used for graft rejection is a mutant that has a slightly higher affinity for B7. So the one that's used for rheumatoid arthritis and psoriasis is called abatacept. The one that's used in transplants is called belatacept. They're both basically the same thing. They're CTLA4-Ig fusion proteins, and they induce co-stimulatory blockade. And it says ongoing trials in transplantation. It's actually now approved for acute graft rejection. And John will tell you more about it. Tregs use B7. So if you treat patients with a B7 antagonist, will you deplete Tregs and make the disease worse? This has been a theoretical concern. It hasn't happened, as far as we can tell, in people at the doses at which they're treated. However, you block signal 2, you'll get increased susceptibility to infection. Every time you muck around with the host response, you're going to get increased susceptibility to infection. So there are all kinds of dire warnings not to combine this with anti-cytokine antibodies. So you have to be careful using, this is basically an immune suppressant. The other side of I'm going to skip that because that's a theoretical notion. The other side is this, which is you block CTLA4 with an antibody against CTLA4. So this is not CTLA4-Ig, this is anti-CTLA4 antibody. And last year it was approved for the treatment of metastatic melanoma. If you treat a patient with anti-CTLA4 antibody, what's the expected side effect? What's the function of CTLA4? Self-tolerance. You treat the patient with anti-CTLA4, they'll get autoimmune disease. And they do. They get inflammatory. That's the formal experimental proof in a clinical trial that CTLA4 maintains self-tolerance in humans. It's unfortunate proof, but it's the proof. Usually we know how to control the inflammatory reaction. Sometimes they can be pretty serious. It turns out the data for PD-1 have just been coming out. Well, I'm not sure how much of it is published. I've seen some of it, but I've also seen the Hopkins group talk about anti-PD-1 clinical trials, and the results are absolutely knock your socks off. So they are really impressive in multiple cancers, and the toxicity is much less. Probably because PD-1 doesn't control early. It's less of it controls late in the response. But blocking PD-1, the principle is the same. You're taking the brakes off. the immune response. That's really the principle. And this is obvious. And I just want to sort of wind up this part by pointing out that there is an emerging paradigm in cancer immunotherapy. So far in cancer immunotherapy, we've tried to vaccinate. We've tried to boost the response. What we are learning is that as, or perhaps even more effective, it is to let the host do the responding. All you need to do is to take the brakes off. You remove the brakes. And this is a really new concept. Many people in the field think that this is going to become standard of care for tumors, that you have an anti-PD-1 or anti-CTLA-4 antibody to remove the brakes and a kinase inhibitor to hit the tumor directly. And that combination, so combinations of BRAF inhibitors and anti-PD-1 in metastatic melanoma, the trials are already underway. And it's possibly the most exciting advance in cancer immunotherapy in the last 10 or 20 years, is this concept of taking the brakes off. And so I just want everyone to understand. What's also particularly satisfying about this is that this has come entirely based on fundamental research. This is understanding the science and the biology and translating it to people. And so when I say this is why we do what we do, I mean, this is really, to me, this is one of the most glorious examples of going through systematic, hardcore basic science and figuring out how to apply it. Okay, so I've gone through all of that. I'm not going to rehash it. I just want to point out that the emphasis very clearly in my talk has been on co-stimulators of this one family. There are lots of proteins of the TNF receptor family that have been claimed to have co-stimulatory activity. None of them has been knocked out and so on and achieved the same level of conviction as the B7-CD28. So they're out there. Take that for what it's worth. There are lots of people who reported, lots of them. I personally think B7-CD28 are the most important for initiating an immune response, but there may be others that also contribute. I just want to end up with one more slide, which is this tumor. This is the idea that when you infect a mouse, for instance, the number of T cells expands and then contracts quite rapidly. There are two interesting points that are made here. One interesting point, the expansion of CD28s is 100-fold greater than the expansion of the expansion of CD8s is 100-fold greater than the expansion of CD4s. And we still don't know the mechanism of that. But I do want to emphasize one thing, which I'm going to skip that. That's pretty obvious and it's straightforward, which is the magnitude of the expansion. At the height of, in a mouse, 1 in 100,000 T cells are specific for a virus. At the peak of the infection in the mouse, as many as 1 in 3 CD8 cells are specific for that virus. In humans with infectious mono, as many as 10% of circulating CD8 T cells are EBV specific. This is prodigious. It's the most extraordinary expansion that the system goes through. And then it all kind of calms down and contracts and everything returns to more or less baseline with just a few memory cells left. And it's all controlled within a couple of weeks. I'm not going to go into much detail. I'll tell you about interleukin-2 later on. I'll skip that and just end with this, that we started with T cells going through lymph nodes and finding antigen. We've talked about what signals it takes to activate them. I've gone through just the first step of the activation, which is clonal expansion, and then Andy is going to pick up from here and go through the next step in the process, which is the generation of effector T cells. 5-minute break.